Hello, and welcome back to Introduction to Genetics and Evolution. In the previous video, we talked about trying to identify differences between species, focusing on humans and chimps in particular, that may have spread by the action of natural selection, and contrasting those with ones that may have spread by drift. There are several challenges associated with this. The way we address these challenges is by contrasting non-synonymous changes and synonymous changes. So this is looking specifically at protein coding genes. So non-synonymous differences are the ones, nucleotide differences, that will change the amino acid that goes into the final protein. So these non-synonymous differences may be positively selected, they may be advantageous or good, and therefore they spread quickly within one of the species. They may be negatively selected or under purifying selection. That means that they're basically like bad mutations. So they arise, they may stick around for a little bit, but largely selection is trying to eliminate them. Or they may be neutral. They may arise, they may change the protein, but not particularly change anything about fitness. In that case, they bebop around within the population. In contrast, synonymous changes do not affect the final amino acid. So those are considered to be largely, though not completely, neutral. So what we do when we're trying to infer the action of natural selection is to try to scale the number of non-synonymous changes, which is sort of the experimental group, with the number of synonymous changes. These are the ones that are thought to accumulate neutrally. So again, we can use them to scale for possible mutation rate differences at different genes. So we're looking for the ratio of non-synonymous to synonymous differences to estimate this. Specifically in this case, we'll focus on the measure referred to as dn over ds. Now, these are not just number of non-synonymous differences, but dn is the number of non-synonymous changes per non-synonymous site. Okay? ds is number of synonymous changes per synonymous site. You may remember I mentioned that the second position of every codon is always non-synonymous. So there's no opportunity for any change there to be synonymous. So we have to use this kind of extra scaling even within our measures of what's happening in terms of non-synonymous and what's happening in terms of synonymous. Well, let's try this out. So what we have here are two DNA sequences, each with 4 codons. Okay? So let's say this top one is human, just as an example. The bottom was chimp. Now, so these are the sequences. These are the DNA sequences or the RNA. These are the resulting amino acids that would come from them. So we see there is an amino acid difference here in that last one, and it's probably resulting from this nucleotide difference. These two other variable sites are not actually changing the amino acids because we see that this one's ACT, this one's ACG, but they both have threonine. So what we're going to do is we're going to walk through site by site. So we have 12 different nucleotide sites here. We want to do two things. We want to classify each site as being something that could potentially cause a non-synonymous difference or could potentially cause a synonymous difference. We're going to walk through site by site. We're going to tally it up. We want to see how many potential synonymous differences we could have, how many potential non-synonymous differences we have. And then we'll contrast that with the number of actual synonymous differences and non-synonymous differences. So let's start walking through. So looking at ACT versus ACG, what would happen in terms of this first site here? What would happen if let's start with this sequence. Let's say that we started with ACT, here it is in our codon table. If we change that first base to anything else, if it became CCT, TCT, or GCT, what would happen? Well, if we did any of those things, it would actually change the amino acid. TCT is a serine, CCT is a proline, GCT is alanine. So we classify this first site as a non-synonymous site. Okay? Because any change from A gives a different amino acid. This is a clear non-synonymous site. As I mentioned before, all second positions are non-synonymous sites. Any second position change will change the amino acid. So this is definitely a non-synonymous site. And the third position in this case is a synonymous site. Because any change here, if it's T, G, it doesn't matter what it is. If you're starting with AC, any change there will not affect the amino acid. So that is a clear synonymous site. So what we do is we tally up for this entire sequence how many potential synonymous changes were there, how many potential non-synonymous changes were there. And what we see is the total number of synonymous sites is 4. It's just the total of these numbers. The total number of non-synonymous sites is 8. Now you may be wondering, what about bases where some changes affect amino acid and some changes don't? So one example would be like this one, TTT. If you started with a TTT, which is a phenylalanine, you could change from TTT to TTC, and that's still phenylalanine. But if you change that third nucleotide to a G or an A, it becomes a leucine. So that's a little bit trickier how you deal with those, but it's not so bad. Essentially what you do is if you're starting with TTG, your third position has a 1/3 probability of being synonymous and a 2/3 probability of changing to a non-synonymous. So what you do is you add both those totals into those amounts. So in the synonymous column, where I had like the zeros and then the 1, you just put 1/3. In the non-synonymous columns where I had 1, 1, 0, you just make that one a 2/3. Okay, so it's fairly straightforward. I'm not going to give you problems like that here in this online course, but I just wanted you to know what would happen in that case. So again, we have this 4 synonymous sites, 8 non-synonymous sites. When we look at the changes we actually have 2 synonymous changes. This is one synonymous change, this is the other, and we have one non-synonymous change, which is that one right there. Now we're not necessarily showing a direction. We're not saying it changed from C to T. It may have changed from T to C. We don't actually know, but we're just calling these differences. Now, what we want to do is we want dn over ds. dn is non-synonymous changes over non-synonymous sites. ds is synonymous changes over synonymous sites. So let's put those together. dn is non-synonymous changes, which is 1 over non-synonymous sites, which is 8. So 1/8 or 0.125. ds is 2 out of 4 synonymous changes over synonymous sites, or 0.5. So dn/ds would just be this number divided by that number, which would be, in this case, 0.25, which is a fairly typical dn/ds value that you might find. Now, you might wonder, what does this mean? So what does any dn/ds value mean? Well, this is estimating how much non-neutral or non-synonymous evolution has happened relative to neutral or synonymous evolution. Well, if a gene is evolving truly neutrally, if it really just didn't matter what differences you saw, if anything was equally okay in terms of fitness, we expect a dn/ds value close to 1, right? And that's not at all what we saw, but we expect the dn/ds value close to 1. So this is saying there's no selection on non-synonymous changes. There's no bad, there's no good, that basically they're just like the neutral ones. In that case, you would actually have more non-synonymous changes than synonymous changes, but since you have more non-synonymous sites, it factors that out. So this would be what's happening in terms of neutrality. Another possibility is that you have dn/ds well below 1. That means that changes have been constrained. This is very typical. In this case, most non-synonymous changes that arise have been selected against. As we mentioned before with that flu virus example, this is pretty typical. You can also have dn/ds greater than 1. This is less common, but quite interesting. What happens in this case is you're having very rapid changes. That basically, within a single gene, you're having multiple non-synonymous changes favored by the action of natural selection. That's really cool when you see that, and that is very strongly indicative of strong, recurrent, positive selection. So let me show you a plot. Here's dn/ds values between humans and chimps. So the average dn/ds across the genome is 0.23. So there you go, that 0.25 we saw was fairly typical. There are 585 genes out of 13,000 that were tested that have a dn/ds value greater than 1. So these are the ones that are undergoing recurrent positive selection. So that's very exciting. These are often genes involved in resistance to parasites or fertilization. These are ones that you would expect to undergo rapid recurrent evolution. And this figure just shows you a sliding gene window, and you see these little peaks here. Like, for example, the epidermal differentiation complex seems to be associated with a very high dn/ds value. Well, let me give you one to try yourselves here in an in-video quiz. So try this. This is the ASPM gene, which affects brain size in humans. And I have here a set of sequences, and I want you to go through and calculate what the dn/ds value would be. Okay, well, let's check this out and see what you guys got. So what we want here is we want to look at non-synonymous sites, synonymous sites. So let's do the sites, and then we'll look at the actual changes. Now the changes, I'll go ahead and just highlight those so you can see them. These are the bases that are actually different in each case. I think that's it. Okay, so when you look at non-synonymous sites, what I did just to make it simple, these are all sort of typical codons where the first and second position are non-synonymous, and third position is synonymous. Okay, so this would be 1, 1, 0, 0, 0, 1, 1, 1, 0, 0, 0, 1, 1, 0, 0, 0, 1, 0, 0, 0. So these are all typical ones. You can look up the actual codons there. GTA, if you change it to GTC or GTG or anything like that, it's all the same. If you change the middle one, it's always non-synonymous. And these first ones, the ones I picked, are ones that if you change that first base, it would actually change the amino acid. So these are the sites. Now we look at the actual changes. Well, actually, let's total these up first. In terms of non-synonymous sites, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. For synonymous, there's 1, 2, 3, 4, 5. So we have 5 synonymous sites, 10 non-synonymous sites. When we look at changes, we have 2, this one and this one, that are non-synonymous. And synonymous, we have 1. Okay? So our dn over ds, so dn will be equal to non-synonymous changes over non-synonymous sites. So that would be 2 over 10, which is equal to 0.2. Our ds is equal to 1 over 5, which is equal to 0.2. So our dn over ds is very easy to calculate in this case. It would be 0.2 divided by 0.2, or it would be 1. So what does that tell you? And this comes back to the question of what a dnds value really means. The real dnds for ASPM is actually 0.9, so that 1 was actually not too far off. So is it likely this gene is actually evolving neutrally? This gene that affects brain size in humans, is it very likely that any amino acid change to this gene has no fitness effect whatsoever? You can change it in any way, shape, or form. No, it's really very unlikely that any amino acid change there really don't matter. What's probably happening instead is we're having a combination of both constraint and rapid evolution within the same gene. That there are some nucleotide changes there that are being rapidly selected out, but there are some that are actually beneficial and they're going through. When you have both of those together, your dnds value is taking this kind of average across the whole gene. So when you have constraint and rapid evolution together, it's kind of hard to tell what's going on, and this muddies how you can do a single generalization about a gene as a whole. And some people will actually break up the gene and look at sections of it independently. But this comes back to this broader question of what these dnds values mean. That essentially, if you have a dnds value of 1, that doesn't mean the gene is actually evolving neutrally. It means you cannot reject neutrality. And in fact, realistically, it's unlikely that almost any protein coding gene would be really evolving totally neutrally. It's very improbable. It could happen, but it's very improbable. So if you have dnds less than 1, you may still have some adaptive changes in there, but you have, importantly, lots of constraint. That most changes that come up there are bad, and they're taken out. Most amino acid changes are disfavored. Similarly, if you have dnds greater than 1, then you definitely have selection driving rapid change. You probably have some constraint as well. Probably not any possible change is good, but probably a subset of them are bad, but you have nonetheless had multiple amino acid changes favored. So what's happening here is you're basically looking at an average of an evolutionary process when you're looking at this single dnds value. So you can tell that there's been a lot of constraint or a lot of rapid evolution, or you just can't tell. That's really what it comes down to. Well, this is not entirely satisfying, as you can tell. We basically need another test, because dnds can end up being a little bit too conservative, especially if you're looking for those adaptive amino acid changes, that high dnds value. You'll have way too many false negatives. So in the next video, we'll look at a test that is a little bit better at catching these kinds of adaptive amino acid changes. It's referred to as the McDonald-Crichton test. Hope you'll join us. Thank you. Hello, and welcome back to Introduction to Genetics and Evolution. In the previous video, we talked about looking for evidence for natural selection at individual genes, and trying to focus on differences, say, between humans and chimps, and infer whether these differences we see may have been the result in part from the action of natural selection versus genetic drift. dnds test is useful in this regard. This is looking at the number of non-synonymous changes per non-synonymous site divided by synonymous changes per synonymous site. The advantage of scaling with the synonymous changes is these are expected to be neutral, whereas the non-synonymous changes may be advantageous, may be neutral, or may be detrimental. The problem, though, is dnds can sometimes mix up constraint with adaptive evolution and just call it neutral. So this is a little bit of a challenge, and dnds in general is thought to be fairly conservative when looking for adaptive amino acids. When I say conservative, I mean that there's too many false negatives. There's too many times when you fail to see positive selection that actually has potentially operated in the evolutionary history of a particular gene. Now, we can leverage some aspect of neutral theory that we haven't talked about yet, and that is that we predict the ratio of non-synonymous to synonymous changes should be roughly constant through time, right? That the ratio that you see among individuals within species should be roughly similar to the ratio you see between species. You may be wondering, why should this be constant through time? Well, there's an assumption about the neutral theory that most non-deleterious, non-synonymous mutations are neutral. Basically, those mutations that you see within a species that are just segregating around, they are at some frequency, those tend to be neutral because the bad ones are eliminated, the advantageous ones spread. So you tend to see a lot of these neutral ones. So these non-deleterious, non-synonymous mutations will behave like synonymous mutations, and they'll rise at some sort of constant rate, and they'll fix with about the same probability as a synonymous change. That, this idea of the allele frequency should predict 1 over 2n. Now, two fellows, John McDonald and Marty Kreitman, suggested this test, which has subsequently been named after them. They didn't call it the McDonald-Kreitman test. They just named it a test. Now, they proposed that you can test for selection by seeing if this ratio is constant within species and between species. The importance here is this within species is looking at the present day. When we're taking a snapshot of a population right now, what do we see in terms of non-synonymous variation, synonymous variation? The between species indicates historical changes, what's happened over the evolutionary history. So what they do is they align sets of sequences and identify the variable nucleotide sites. And they basically just have this 4-cell test where they identify each difference as a non-synonymous difference within species, a synonymous difference within species, a non-synonymous difference between species, or a synonymous difference. So let's try this. That was a lot of words. Let's try it in practice. So here we have an example. Let's say we have 4 individuals of species 1 and 1 individual of species 2. For the sake of argument, let's say species 1 are humans and species 2 are chimps. Now here's our codon table again. And what we're looking for are these 2 parallel categories. First, what is fixed between. So these are differences between species 1 and species 2. We have polymorphism within. So basically what is variable within, in this case, species 1. And then we can also categorize any of these differences as being non-synonymous, or amino acid changing, or synonymous, where they don't change them. So what we do is we just walk through. In this case, we're not looking per site. We're actually just looking overall. So where are the differences between species here? Well, here is a difference, or within or between species. So there's a difference here between species. This is within species. This is between species. This is within species. This is within species. So we have an amalgam here. We have some within species differences, some between species differences. Okay, now let's walk through them. So this first site here, this is a non-synonymous change. As you can see here, a CT anything to ATG, which is methionine. That is a non-synonymous difference, and this is a non-synonymous difference between species. So that goes in this cell. We skip the next one because we're not going per site. We're just going to skip the next one and go to the third nucleotide site. In this case, we have a difference within species, but they all are making leucine. So this would be polymorphic within, right, and synonymous. So let's jump now over a little bit. This one right here is a difference between species, but again, AC anything is threonine, so this is a synonymous but between species difference. Jump over a little bit. This is a within species difference, but second position. Second position is always non-synonymous, so this would be non-synonymous within. And the last one is over here, ACC or ACA, they're both threonine, so this would be another polymorphic within species, because the variation is just in species 1. Species 2 has one of the ones from species 1, and it is synonymous. So we basically tally these things up, and we have these 4 numbers that come out. We'll just call them A, B, C, and D for the sake of argument. If all non-synonymous differences are neutral, okay? If all non-synonymous differences are neutral, then we expect A divided by C, this ratio that you see between species, this is essentially reflecting the historical, should be equal to the ratio of B over D, what you see within species, or the present day. In contrast, if some, or especially if there's a lot of non-synonymous differences between species that were advantageous and selected, let's say something helped us humans be able to digest things that are unique to our diet better, then this ratio should be greater than this ratio. That the A over C should be greater than B over D. Okay? That what's happened then is these differences will have spread within humans. They are changing the amino acid variation. Since they spread within humans, they are now a between species difference. They're not something that's neutral, that's just kind of hanging out in the population. Okay? So let's look at a couple of examples. Here's some actual data from a few genes. Here's SEMP1. That enables cellular survival under low oxygen. This is a case where we see a large difference in terms of non-synonymous over synonymous between species relative to within species, this would be a good candidate for positive selection. That there's been some sort of adaptive difference between humans and chimps. In contrast, this one, CIAS1, which affects autoinflammatory response, that one is pretty similar between the two. So this is a case where we can't reject neutrality. The ratio here, 5/10 is equal to 1/2. Now, what about the opposite? This is what we haven't talked about. What happens when, instead of being greater between species or equal, what happens if it's the other way? So here's the gene AGT. The mutations in AGT affect hypertension, or associated with coronary artery disease. What happens if you have a high number of non-synonymous differences among humans, a higher ratio of non-synonymous differences among humans than between humans and chimps? What do you think that might mean? So now we have to think about what kind of variation would do this. So what kind of variation persists for a long time but doesn't fix? Well, the answer in this case is unfit recessive alleles. So let me show you a picture. And this is a picture you saw before. Let's say over here on the x-axis is time in generations, and on the y-axis is allele frequency. Now let's imagine hypothetically that you had an allele that was actually bad, but it was recessive. What you might see something that looks like this. We saw this actually in previous videos when we talked about the action of natural selection, that it's actually kind of hard to completely get rid of these recessive bad alleles. These bad alleles can stick around for a long time because what's happening here, let's say for example the bad one is little a, but then the good ones are big A, and big A, little a, right? What happens is it gets very rare. Almost all the little a's are present in the heterozygous form. So they're basically masked, and selection isn't getting rid of them very well. Only the very small number of little a's are actually being eliminated by natural selection. So the better dominant allele never quite gets to fixation. Now, a lot of diseases, as I mentioned way back when, tend to be recessive. And as a result, these mutations causing disease will stick around within populations, but never fix, and they eventually do get lost, but it takes a very long time for them to get lost. So this is variation that you'll see that will stick around within species, and it affects this within species numbers, but does not affect the between species numbers. And these diseases will tend to be associated with non-synonymous bad variation. So this is what causes that essentially, there's the ratios again we talked about, between species non-synonymous over synonymous versus within species non-synonymous over synonymous. That if all non-synonymous differences were neutral, we expect these ratios to be similar. If some of these non-synonymous differences were advantageous and selected, then we expect this ratio, A over C, to be greater than this ratio, B over D, right? Because the advantageous variance would spread. They would become between species non-synonymous variation. In contrast, if you have maladaptive non-synonymous differences, they will persist within species and never be converted to between species differences. In that case, A over C will be less than B over D. That's what will happen if you have diseases lingering around. So, looking at the McDonald-Kreitman test, we have this A over C equals B over D. That's a case where you can't reject neutrality. Again, it doesn't mean it's neutral. It can be a mix, but you can't reject it. If A over C is greater than B over D, then we have increased non-synonymous changes between species. We have more non-synonymous changes than we expected between species relative to within. And that is indicative of positive selection or advantageous variation. So you're running. In contrast, if A over C is less than B over D, then you have decreased non-synonymous changes between species. What that means is you have some variation that's lingering within species and doesn't quite get fixed. That is often the result of negative selection. There are other possibilities. Things like overdominance that we talked about earlier will also do this same sort of pattern, but it's thought that overdominance is not especially common, so a lot of this is going to be in this boat of negative selection or bad variation sticking around. So what happens when we look at the genome? I showed you a couple of examples. This is looking at a lot of genes studied across, or differences between humans and chimps. The blue marked genes are ones that have McDonald-Kreitman tests results indicative of positive selection. The red ones are indicative of negative selection. You notice there's a lot more red out there than blue. But again, we know that most mutations are bad. We know that most bad mutations are recessive. Therefore, it's going to make it so they're just kind of lingering around. So instead of just looking at this figure, let me give you some of the numbers. The positively selected genes from that particular study I was just showing you, there are about 304. Again, what are they associated with? Same sorts of things as the high DNDS values. immunity protein genes, gamete formation ones, also some for sensory perception. In terms of negatively selected ones, many of these are involved in cytoskeletal formation, and they're often associated with diseases like muscular dystrophy, cardiovascular disease, etc. So let me give you one to try here, so you can have a little bit of practice within the video before you even try any of the practice problems. So this is a hypothetical sequence for ARSD. This is something that controls arsenic resistance. Okay, so I want you to go through here and calculate and interpret the McDonald-Kreitman test. So you may want to pause the video before you actually get to the in-video quiz. Okay, well, I hope that wasn't too difficult for you. Let's go ahead and take a look. So what I'm going to do is I'm going to highlight things in blue and red first. So the blue ones here are indicating differences between species. So this is comparing humans to chimp. The blue ones are indicating differences between species. The red are indicating sites that are variable within species, just to make it a little bit easier to see. So let's look at those. When we're looking between species, we have one non-synonymous difference. That's this one right here. This is non-synonymous. And we have two synonymous differences between species. Right? So this is a synonymous difference here. This is a synonymous difference here. When we're looking within species, what do we see? We have two non-synonymous. This one is non-synonymous. Remember, always the second site is non-synonymous. This one's also non-synonymous. And this one right here is synonymous. So one synonymous within species. So our ratio is 1 to 2 versus 2 to 1. So again, which one is bigger? In this case, we see it's bigger in the within species category. That this is the result in the direction of negative selection, that there is potentially bad variation sticking around within species, not being eliminated. So let's recap real quickly. Here's the McDonald-Kreitman test. Again, if A over C, A being the between species non-synonymous divided by the between species synonymous for C, is equal to B over D, the same for within species, you can't reject neutrality. Does not mean that it is neutral. If it's greater between species than within species, then you have increased non-synonymous changes, that interesting positive selection, right? In contrast, if you have A over C less than B over D, you have decreased. You've been holding back the non-synonymous changes. They're staying within species and not converting to differences between. So that is negative selection. Now, I've shown you the DNDS test and I've shown you the McDonald-Kreitman test. There are other metrics out there. There are many that also exist that we won't use in class. One popular one is referred to as Tajima's D. That uses the frequencies of alleles to test for recent selection or recent changes in population size, as an example. But importantly, these are implemented to understand what's going on. There's two general approaches for studying. One is to scan the whole genome using these metrics and look for possible selection and then try to interpret it. Right? So that's like the picture I showed you there showing all the red and blue labeled genes. That's when you're just looking at everything and trying to interpret how much positive selection versus negative selection is there in the genome. The other is to look at candidate genes, to pick out the subset of genes that you know affect brain size or affect speech, and look for the signatures of selection at those, or maybe even individual genes. Now, both of these are active areas of research, and both of them have given us some clues as to what's been important in our divergence or the divergence of any other species that have been studied. So closing out, what makes humans special? Well, these DNA sequence-based tests have shown that, again, hundreds of genes have undergone selection to change. And a lot of that seems to be, when I say selection to change, I'm referring to this positive selection, these adaptive variants. More genes are under some selection to stay the same, that basically non-synonymous changes are bad, and therefore they're held back. They don't ever become differences between species, but may linger as differences within species. And therefore, some of these bad alleles are sticking around. And this also makes it so a lot of our sequence is more similar to chimpanzees than if we were evolving purely neutrally. But importantly, let me just close with this. There's no single gene change that made us what we are today. Yet, we're not as different from chimpanzees genetically as we might like to think. Well, I hope you found this interesting. Thank you for joining.